This video is about molecular systematics and DNA barcoding. So let's start learning. Modern taxonomy focuses on not only the classification of organisms but also their phylogeny. Phylogeny is the study of relationships among different groups of organisms and their evolutionary development. Phylogeny attempts to trace the evolutionary history of all life on the planet. It is based on the phylogenetic hypothesis that all living organisms share a common ancestor. Bacteria, archaea, and eukarya. Then molecular systematics. We are going to learn about molecular systematics. Molecular systematics. Molecular systematics is a study of systematics using molecular data or study of molecules. Study of systematics at molecular level. It works as the association of systematics and bioinformatics, so that molecular systematics is also called computational systematics. Three main aim of molecular systematics: reconstruction of phylogenetic relationships of organisms, studies of population structure including geographic variation, mating systems, heterozygosity and individual relatedness, then identification of species boundaries including hybridization, then principles of molecular systematics. Classification is done based on the similarity and dissimilarity in molecular level. So we learned three main aims: reconstruction of phylogenetic relationships of organisms, studies of population structure including geographic variation, mating systems, heterozygosity and individual relatedness, then identification of species boundaries including hybridization, then principles of molecular systematics. Classification is done based on the similarity and dissimilarity in molecular level. Nucleotide sequence and protein sequence of different organisms are compared. Closely associated organisms should have proteins with similar amino acid sequences or genes with similar nucleotide sequences. This is the structure of some unaligned sequences. The sequence alignment is shown here. Histone H1, non-conserved amino acids, conservative portions, non-conservative portions, etc. shown. Molecular systematics use variety of techniques to derive phylogenetic tree. Polymerase chain reaction (PCR) is used to investigate variations of DNA on a large scale. DNA amplification is also fundamental to new approaches to DNA fingerprinting. Molecular clocks is used to predict past and future divergence in genes. This theory claimed that molecular change is sufficiently constant to determine how current genetic lineages branch off from common ancestors and to determine when the branching occurred. Genetic markers are used to interfere inferences. Genetic markers are used to inferences about relationship between environment and morphology as well as physiology and behavior. So protein taxonomy introduced by Crick. A species can be differentiated on the basis of amino acid sequences in the protein of an organism. Molecular taxonomy divided into macromolecular and micromolecular taxonomy. First, we are going to learn about macromolecular taxonomy. Macromolecular taxonomy stress on the distribution and biosynthesis interrelationships of small molecular weight compounds such as free amino acids, terpenes, flavonoids. This approach is especially useful in resolving systematic problems where hybridization has been a factor. Then later we are going to learn about macromolecular taxonomy. Macromolecular taxonomy concerned with polymeric molecules like DNA, RNA, polysaccharides and proteins. DNA is an excellent tool for phylogenetic studies. Enzymes and proteins are also studied under molecular systematics to find out the phylogenetic relationship between different organisms. So we learned macromolecular taxonomy and along with that micromolecular taxonomy. Here is a mistake. It's macro and micro. Macro in the sense large molecule, polymeric molecule like DNA, RNA, polysaccharides and proteins. Micro in the sense free amino acids, terpenes, flavonoids, small molecular weight compounds. Okay. The micromolecular, first one is a micromolecular. Don't confuse between these two. Micro, small, that means small molecular weight compounds we are analyzing and finding their biosynthetic interrelationships. For example, free amino acids, terpenes, flavonoids. This approach is especially useful in resolving systematic problems where hybridization has been a factor. Hybridization has a influence in this process of micromolecular taxonomy. Then macromolecular taxonomy is concerned with polymeric molecules like DNA, RNA, polysaccharides and proteins. So DNA is an excellent tool for phylogenetic studies. Enzymes and proteins are also studied under molecular systematics to find out the phylogenetic relationship between different organisms. some major applications: comparison of homologous sequences of genes using sequence alignment techniques to identify similarity, then DNA fingerprinting and DNA barcoding. Then blood sample is here. DNA isolated from blood sample, DNA subject to restriction digestion. So blood sample is collected, DNA isolated from blood sample, later DNA subject to restriction digestion. Along with that, fragments of DNA are separated by electrophoresis, later transfer of DNA fragments to the membrane. Southern blotting is performing here. Then hybridization with radioactive probe, later excess radioactive probe is washed, developing the X-ray film to detect the radioactive probe. DNA pattern is compared with pattern of known subjects. Once again, we are going to look at this picture. First, blood sample is collected, DNA isolated from blood sample, then DNA subject to the restriction digestion is taking place, then fragments of DNA are separated by electrophoresis. Along with that, transfer of DNA fragments to the membrane. Southern blotting is also performed. Later, hybridization with radioactive probe is happening. Along with that, excess radioactive probe is washed off, then developing the X-ray film to detect the radioactive probe. That is also happening. Later, DNA pattern is compared with patterns of non-subjects. Then DNA barcoding. DNA barcoding is the standardized approach to identify plants. We just started learning DNA barcoding just by completing molecular taxonomy. We are going to learn about DNA barcoding. We just mentioned one application of this molecular taxonomy. One of them is the DNA barcoding. So DNA barcoding is the standardized approach to identify plants and animals by minimal sequence of DNA. DNA barcode, short sequence taken from a standardized portion of the genome that is used to identify the species ranging from 400 to 800 species. DNA barcoding is the standardized approach to identify plants and animals by minimal sequence of DNA. DNA barcode, short sequence taken from a standardized portion of the genome that is used to identify the species ranging from 400 to 800 species. Sequence of unknown species can be matched from submitted sequence in Gene Bank BLAST. So BLAST, Basic Local Alignment Search Tool, is a web program for searching the closely related sequence. If you guys try to perform BLAST analysis, we know how multiple sequence alignment is performed here. Just we are going to align one particular sequence of this organism or whatever it may be, that organism, into the large database, and we are performing analysis, alignment to choose the appropriate one, which is to a great extent similar to the one we are searching for. So that is BLAST is a basic local alignment search tool. Basic local alignment search tool. Basic local alignment search tool. Something similar to cluster analysis. These are some bioinformatics tools. I prefer to search about them. There are also some other bioinformatics tools like PhyloDraw, which help to draw phylogenetic trees. Cluster Omega is Cluster Omega is also a tool which is used to create this alignment, and along with that, some phylogenetic trees are also created with the help of that. So BLAST is a basic local alignment search tool. A web program for searching the closely related sequences. Species DNA barcode. History of DNA barcoding. The use of DNA sequences for species identification has long history, but it received significant attention only after it was formally proposed as DNA barcoding in 2003. Paul Herbert proposed DNA barcoding, and he is considered as father of barcoding. His idea was to generate a unique identification tag for each species based on a short stretch of DNA. Paul Herbert proposed a part of gene called cytochrome C oxidase CO1 as suitable to DNA barcoding. CO1 acts as a universal marker for species level identification in several animal groups. CO1, cytochrome C oxidase 1 by Paul Herbert. Why CO1? Cytochrome oxidase 1 portion. Easily extracted. Mutation rate is fast. Standard region is enough to distinguish closely related species. lack insertion or deletion, greater differences among species copy number, relatively few differences within species, then absence of introns. Here we could see a picture of CO1 DNA segment used for barcoding. Once again, I'm going to read this. Why this is chosen as a marker, universal marker, CO1. Easily extracted, mutation rate is fast, standard region is enough to distinguish closely related species, lack insertion or deletion, greater differences among species copy number, relatively few differences within species, absence of introns. Then DNA segment used for barcoding, prokaryotes there, animals, birds, fishes, fungi. Small regions of mitochondrial CO1 are RNA genes used for species identification, RBCL gene sequence and MATK gene sequences of chloroplast, then ITS region and fungi. Steps involved during DNA barcoding. Specimen collection, that is the first step. Specimen collection, extraction of DNA, amplification of specific DNA segment by using specific primer, sequencing the amplified DNA, compare the resulting sequence against a reference database to find the matching species. Just remember the BLAST program. Earlier we mentioned about that. Then specimen collection. First step was specimen collection. Collecting fresh or dried specimens from plants, animals, fungi, insects, etc. Fresh samples are easier to isolate than dried because dried specimens contain intact DNA. It is more difficult to isolate. So dried specimens are common for plants and fungi collection. During sample collection, do not take more sample than you need. Only small amount of tissue is needed for extraction. Plants leave about 1/8, 1/4 inch diameter piece of animal or fungal tissue. That's enough. So in the case, small invertebrate animals such as insect can be collected as whole and euthanized in a killer jar. Then DNA extraction. DNA extraction is a method to purify DNA using physical or chemical method from a collected sample. In DNA extraction, separating DNA from cell membrane, proteins and other cellular components. DNA extraction technique include organic method, phenol chloroform method or non-organic method, salting out and protein kinase K treatment. Then absorption method, silica gel membrane, amplification of specific DNA segments. The extracted DNA is amplified using polymerase chain reaction or PCR to produce multiple copies of specific DNA sequence. Amplification achieved by 3 steps: denaturation, annealing, and extension. Denaturation: the sample first heated and separating the DNA strand by breaking hydrogen bonds. Then annealing, primer bond to target DNA and initiate polymerization. Then extension phase, DNA polymerase extend the 3' end of each primer along the template. Then sequencing and data analysis. To determine the order of nucleotide bases, sequencing the amplified DNA barcode marker region. So to determine the order of nucleotide bases, sequencing the amplified DNA barcode marker region. Once the whole DNA is sequenced, the result is saved into one unique file format and compare the resulting sequence against reference library bolt to find the matching sequence.